It's a pleasure to be here and to talk about structural biology. In thinking about what to talk about, I decided at the end sort of to go back a little bit to sort of more classical things, and so I'll be talking mostly about how we use molecular dynamics simulations to study protein dynamics. So skipping a little bit out of the framework of AI. I will see you do, as many others, I do machine learning, use machine learning for a lot of tools. I was almost considering talking about disordered proteins, so I was very happy that Martin gave a wonderful introduction and overview of this, and happy that I didn't have to compete with that. I will just say that we have run molecular dynamic simulations of all human disordered proteins and put them in a database if you are tired of looking at the wrong spaghetti that AlphaFold spits out. We've also trained a machine learning model, so you don't even need to run simulations of these things. And we also use a lot of machine learning models to interpret the structures that either you as structural biologists or machine learning methods spit out. But I won't talk about that. Instead, as I said, I'll go back a little bit to sort of the roots of what I've been working on for the last many years of using molecular simulations as a tool jointly with experimental data to shed light on what molecules look like and what they do. And before I get to that, I will just say that if there's anything that I talk about that is of interest to you, we try to put out everything as openly as quickly as possible. There will be a little bit of unpublished stuff that I haven't been able to put out yet, but basically there will be a preprint and a GitHub repository for every paper that we have out there, so you should be able to play with all the data and all the code. So we've seen some version of this with a much more interesting animal earlier, but I like to show horses, and so in the ideal case, we want an experiment that can give us images like this. These are images that have high spatial resolution and at the same time high temporal resolution. Right? So we want to be able to see objects as they move rather than just the end state, which can of course also sometimes be interesting. Right? The technique here is one you're all familiar with, right? It is photography, and in this case used together with stroboscopic lights, sort of to get some high resolution snapshots along the way of the process. Most of the images that we worked with look something like this, and for those who heard me speak before, you might know why I showed you a picture of a swimming pool, and if not, I will just say that this picture has sort of, in many ways, transformed the way I think about how we use experimental data. So as you can see, this is a picture of a swimming pool. It looks blurry to the people at the back, but it also looks blurry to the people at the front. It also looks weird. One of the things when I saw this hanging on the wall of a friend that struck me was something with the light that was odd. And the odd thing about the light, I then realized that there appears to be sunlight coming both from these windows and then these windows over here. And knowing something about the solar system, right? That's unusual if the sun can be in two places at the same time. And the reason for this is that this is taken again with photography, but with what's called a pinhole camera, right, which has a very small hole, and so it only lets a few photons in at the time. And the way you compensate for that is you have a very long exposure time, in this case, exposure time of a day. Right, so this professional photographer, so Peterson, put down the camera, press the shutter, and then expose the picture for a day. And so if you are taking images or doing experiments on systems that are dynamic, on time scales that are comparable to or faster than the experimental time scale that you can access, right, you get these kind of artifacts, you might say. In fact, there is even more, I think, interesting artifact, which is that this was done when the swimming pool was open. So there were actually people swimming back and forth in these lanes during the day. So if you care about, what does a swimming pool on average look like, this is the beautiful experiment. If you care about, how the sun moves, right, you can probably work out backwards maybe something about, the sun can be in multiple locations. If you care about the dynamics of, swimmers, right, this experiment is completely blind to that. Right? And also just note that, this is true for many experiments, right, that this dynamics, in this case, right, this is not discrete dynamics. It would be very hard to have a lot of these kind of images, or even sort of the individual images, and sort of work out afterwards exactly classes of, well, sometimes the swimmers here, sometimes the swimmers here, right? But this is really continuous dynamics, and that's also true for biomolecules. So we have beautiful experiments They give us a lot of information, but they're often blind to the dynamical processes that we also care about. And so, what we have been working on for many years, and together with many other people in the field, is to take wonderful experimental data recorded in nice infrastructure and combine it with molecular simulation, and also using molecular simulation as an interpretation tool to say, we come with a set of simulations. The simulations give us access to a lot of details. Unfortunately, they're still mostly wrong for many, many reasons that we can talk about, so they're not alone predictive. But then we can use them as an interpretation tool together with the experimental data and come up with models about the motions of systems. And again, just a little bit of a simple case here, but just to show you sort of how wrong you can do if you don't think about dynamics. Right? I work in an NMR lab, and in NMR it's something with spins. So if you have a little coin here, you attach a sort of a vector to it, and you repeat this experiment. There's about 10 to the 16 molecules in an NMR sample. So if you carry the average over 10 to the 16 of your spin up and your spin down, you will get a 0 vector. So if my colleague here, the crowd comes to say, I measured the average orientation, or I measured the orientation of a coin, and it's give me a 0 vector, can you tell me the structure of a coin? Right? What I would normally do when we determine structures is I would move the system around until I find a state that explains the data, right? And this structure will explain the data perfectly, right? It has perfect agreement with a 0z component of a vector, right? But it's clearly the one thing that you very, very, very rarely get out of the system, right? And this, of course, is a little bit oversimplistic, I realize that, but this is basically the assumption that we make when we determine many structures, is that we can represent ensemble and time average data by single structures. And the system is a little bit mobile, you're okay, mostly. If it's highly mobile, like with disordered proteins, of course, clearly you are not okay. So this can, of course, be formalized mathematically. There are many people that have worked in it. We've written a couple of reviews. This top review here is sort of quite technical and has some equations and goes through some of the maths. And this bottom review is sort of more conceptual. So these are a good place to read. But just to sort of say here, if you have some kind of observable that you can measure, right, the way that we would typically use simulations is that we would get out some distribution here. This would be the red thing here. This would be the average. We would say, okay, it doesn't agree with what we measured experimentally. So we'll put out a harmonic potential, we'll force it to agree with it, and we'll get out this distribution here that will fit the experiment within error, and that's great. An alternative approach would be to say, well, we don't trust the simulation perfectly, but we at least think that this is the best bet we have of what nature looks like until we did the experiment. So we'll do a minimal perturbation through the force field prediction that fits the data. And in this case, this means decrease the population of this state, increase the population of this state, and thereby move this calculated average closer to this. And this you can formalize using Bayesian statistics or maximum entropy principles or a number of other mathematical formalisms that are basically the same for doing this. And we've done this in some code that we call Bayesian maximum entropy reweighting. There are some typos here because I changed my slides last minute, but it's basically a tool set that allows you to run molecular dynamics simulation, have some data, you can sort of put it in a coherent framework and get out conformational ensembles, and it's relatively easy to use. So here's just some examples from my group, and other people have used the code for other things. It's pretty easy, and it works with many different types of data that you can calculate from MDE simulations and that you can go measure in the lab. So I'll just give you a couple of examples of this. I'll start by talking about nanodiscs. This used to be a movie when it was on my computer. So now I'll use it instead as a teaching moment, just to remember that if you are looking at a single snapshot of something that's mobile, it's not really representative of what's going on. And I've highlighted here just one lipid molecule, and there's nanodisc that is moving about. We were interested in studying membrane proteins in nanodiscs in solution using neutron and X-ray scattering. But before we did that, we said, well, can we just figure out how they look alone in solution? We were not the first to think of this. There were actually a couple of really nice papers already in the literature. There was a fantastic paper from Roland Wieck's lab that determined the structure of this specific nanodisc. It's shown empty here, but it was full with lipids, and it shows what is basically a round structure. of a nanodisc. It's a disc that sounds round. I mean, at least individual helices, so it's not completely round, but it's basically round. And then there was another study from my colleagues in Copenhagen, Lisa group, using X-ray and neutron scattering, and that showed that you cannot fit the X-ray and neutron scattering with a round structure. It has to be an ellipsoidal structure. So this is a more coarse-grained representation, but we basically show with this paper that you cannot represent the same nanodisc. So this is the same disc, same lipids, same composition with a round structure. Right? So NMR says this thing is round. X-ray and neutron scattering says this is not round, this is ellipsoidal. What's going on? And maybe you don't have to think too hard to realize that they're both true or none of them are true, but we're working with a dynamical system that has shape fluctuations. And it turns out that if you look at it on average by NMR, you get something that is roundish. If you look at it on average by something that cares more about mass distribution, then it looks more ellipsoidal. So we turned to MD simulations. We ran extensive MD simulations of this nanodisc, and we saw ellipsoidal shapes, with something where the long axis can be this direction, and the long axis can be this way. It can sometimes be round. And then we started to compare with the data. This is the comparison with the NMR data. The details are not so important, just to say that we do not agree with all of the NMR data. Molecular dynamics at this time still isn't perfect. So, it's not an amazing tool, but at least it gives us some starting point for interpreting the data. We calculated, again, the X-ray and neutron scattering data, just to show here the blue curve is what we predict from MD. It fits the data pretty well over here, but then it misses the structure over here. This is the NMR structure. Actually, it predicts this sex data quite well, but it misses the structure of this minimum. So the MD doesn't really fit the NMR data. It fits it sort of okay-ish. It doesn't really fit the SAX data, but it fits it sort of okay-ish. But then we have this method that I didn't really tell you about, but that is called the spatial maximum entropy reweighting, which basically allows us to take this MD simulation and massage it in the slightest possible way to fit all the data. And then lo and behold, you get out obviously something that fits the experimental data, fits the NMR data. I'm not showing you that. And now we get out a new distribution of conformations that fits all the data. So on this axis here, this number here means how ellipsoidal is my nanodisc. This down here is sort of the round NMR structure. This up here is the very ellipsoidal X-ray based model. And what you can see is you have a nice distribution of confirmation between these two extremes. Right? So NMR gives you beautiful data. It gives you one way of representing the average structure. Scattering gives you also nice data and gives you another view of this. These two are, if you force them to be, I only want to represent my data with a single structure, you get different views. But if you then integrate it with molecular dynamics, you can really get sort of a more consistent picture that shows both something about the structure and the dynamics. And none of this would have been possible without using both the molecular simulations and the NMR data and the scattering data. So this was sort of a simple case and a small system. I just want to show here some other recent work we've done on a slightly more complicated system, and something that, of course, many of you will be familiar with is something that is difficult to deal with, which is when you have both conformational heterogeneity, so difference in shape, but at the same time also compositional heterogeneity. Right? People that do cryo will know this very well, but if you're looking at molecules that interact and they don't interact very strongly, like your system may not always be completely homogeneous. So I'll talk about how Lindorff-Larsen, who's shown here at the bottom, worked on generating a model of this case, in this case, a very large oligomer. So Tanya Mitak, who will speak tomorrow, has been working on this system for a while, and this protein is called SPOP, and it forms oligomers of various lengths. You can think of the unit here as a dimer of SPOP. So there's a dimer interface that's very strong, and then there's a weaker dimer interface that can form this case of a tetramer. And then it turns out that this interface over here, can still bind another dimer, and it turns out that this dimerization constant is independent of the size of this. So this is an isodesic association model. So you get at any concentration mixtures of dimers and tetramers and hexamers, etc., etc. Right? So you have always a mixture. There's no way you can purify it because this then just re-equilibrates. At some concentration of some estimate of this number, this is a log scale. This is the population of the different species, right? So you can see that you have mostly small species, but then you get also large species, in particular, of course, at high concentration. Now, we are going to look at this with small angle X-ray scattering, and of course scattering is more sensitive to large particles. So while this is the size distribution, this is sort of the intensity distribution you would get from a SACS experiment, which means that if we do a SACS experiment, let's say at 30 or 40 micromolar, we would have to take into account 40 mers up to 60 mers of this. And this is sort of an initial model of what the 60 mer would look like, but this is what we'll get to in a second. So we will have, SACS data, there will be a mixture of states, and the proteins will be dynamic, and we need to sort of interpret all of this at the same time. I won't talk much about the biology of this. You can go talk to Tanya about this. Just to say that, and this will become important for a little bit, this protein recognizes substrates that have all these motifs that are spread out throughout the sequence. And so having a large oligomer with multiple binding motifs, or binding sites, is important for them being able to recognize these distributed motifs. Okay, so this is what the data that Tanya and Macuno reported. So this is a concentration series of small angle X-ray scattering data that we want to interpret now in terms of a conformational ensemble that includes also this compositional heterogeneity. Right? So this is the mess we have to deal with. It's obviously low resolution data, but we have nice crystal structures of what the individual structures look like, so we want to put all of this together in a single model. So we came up with a relatively simple workflow that took a while to refine. And before I tell you how it works, I just want to say that the standard approach that many people take to solve these kind of problems is deconvolution. Like they try to deconvolute the thing into individual space. And this sometimes works if you have a very simple system, but here you basically have to deconvolute it into an infinite number of states, right? That's a hard thing to do. And in general, deconvolution methods require pretty strong assumptions about how you deal with data. So the approach that we take in all of our projects, more or less, is the sort of the opposite. We say we try to represent all the mess of the biology in the model, and then we predict the total amount of data, rather than take the data and try to take it out into pieces, and then fit a simple model to the data. So we really don't like deconvolution methods too much. We really like to take sort of a forward modeling approach. So we want to have a model for what the oligomer distribution looks like. We run molecular dynamic simulations of what all the oligomers look like, including all their dynamics. We combine them together to predict the SACS data. The SACS data that we predict doesn't match the experiment, and then we go through iterations of reweighting where we both refine the model for the self-association and refine the models of the conformations to target this data here. And while this sounds like a very error-prone and overfitting risky project, it turns out actually to work quite well. And so we plug in, an isodesmic constant that will be fitting. We get out a size distribution. We have models of all of these states up to 60 mers, and they're wobbling all of them, right, because we're running molecular dynamics simulations of them. And this is the starting point, and then we refine this model. And just to show you roughly what the fits that we get out of, actually, you can hardly see the differences at these concentrations. Actually, the initial model actually fits the data pretty well. So just taking into account the fact that proteins are mobile and that these proteins self-associate allow you to fit the SACS data pretty well. And then with a little bit of refinement of the ensembles and of the association constant, that of course is a key part because that determines the self-association level, right? We can fit out a KD and we can get a very good fit to the small angle X-ray scattering data, in this kind of global model. So now we have models that can interpret both conformational and compositional heterogeneity. This is again what the 60 mer looks like, but there's of course smaller models. And there's this nice helical shape, at least for this specific construct here. So what kind of things can you see? Well, for example, one question that we had was that these math domains that are important for the substrate binding, are they dangling off this backbone or are they sitting tethered down? So we ran two sets of simulations, one where we tied it down to the backbone and one where we let it be free. And we could very clearly see, this is sort of shown here with these chi-square values, that the fit to the SACS data was just a lot worse if we restrained them as opposed to having them mobile. So although SACS is a low resolution technique, it allows us to see these kind of differences even for a very large system like this. I mentioned that there's these binding motifs that are sort of distributed along the sequence of substrates, so we're also interested in, is this somehow reflected in the distribution of the structures of this receptor molecule? And so we looked at the distribution of the distances between neighboring of these mobile orange domains, which then bind the substrates. And interestingly enough, it turns out that the distance distribution between neighboring binding sites very much varies the distance distribution between neighboring peptides in these substrates. So there looks to be some kind of match between the relative distances in these disordered regions and the relative distances in these folded receptor molecules. And here I'll just highlight one tool that I'm not showing here, but this is we have a very efficient mod for simulating disordered proteins that allow you to simulate any disordered protein on Google Colab in a few minutes. If you care about global properties, if you care about local properties, you should use Martin's code instead. Okay, so in the last few minutes here, I'll switch. This will be pretty unpolished. But I think it's important to remember that when we talk about dynamics, it's really not the same as when we talk about conformational heterogeneity. Dynamics involves time, and heterogeneity is sort of inherently, not, a time property. And this turns out, of course, to be important if you want to understand specific processes or the kinetics involved. Or for some measurements, actually turns out that most NMR measurements and many fluorescence measurements, you really cannot interpret the data without taking the kinetics into account. And so I'll talk about two different things, and this is in this review here, what we call time result experiment, which of course is something that many people are familiar with, and what we call time-dependent experiments, which is a different thing. So time result experiment, right, is typically non-equilibrium experiments where you do something and you want to interpret what's going on. Time-dependent experiments mean that you measure something, but whatever you measure, you cannot actually calculate from a structure. You have to take a structure and the associated dynamics and time scales into account to calculate those observables. So this might be things like an NOE and an NMR, right? It's really not a measure of distance, it's a measure of distance and dynamics. Or it might be an NMR relaxation, or it might be FET, right? FET efficiency is not something that's a function just of distances or angles, it's a function of the mobility on the time scales of the fluorescence experiment. So we've been developing approaches, and what we've done so far is ultra simple, but we're working on this at the moment to look at time-resolved crystallography and ligand binding experiments. But what I'll talk about now is really sort of the very basic idea of how one might use molecular simulations to interpret a time result, in this case, a temperature jump sex experiment. And what I'll show is synthetic data, just because then we know what the truth is, and this is work by Simone Carl. And the input would be that you would have some kind of time resolved sex experiment, we want to interpret it. And here one key point that I think a lot of people like me have missed up until recently is that really we should not think about modeling the kinetics in these kind of problems. That's the simplest thing that you get from the experiment. Right? When you do a time-resolved experiment, reading out the timescales is the easy part. Right? The hard part is figuring out what happens at the molecular level, right? What structures are you seeing, what are the motions. So we want to study slow processes. That's very hard with molecular simulations. So we want to be able to use various tricks, including enhanced sampling methods, and those inherently mess up the timescales. So the idea is that we'll use the simulations to represent the conformational free energy landscapes, and then we will use the experiments to give us back timescales and structural information that we'll model together. So the other input, and I'll show you a little bit more detail, will be free energy landscapes along certain reaction coordinates. This is something that we as a field have been able to do for a while, calculate. And again, they will be good, but they will not be perfect. And then we'll do an analysis that's similar to this Bayesian maximum entropy method that I talked about, but we'll have somehow to include the time in this, right? Because now we're looking not just at a single conformational average, but as a conformational average that's evolving over time. And what I'll talk to you about in a second is how we can model this as diffusive processes on free energy landscapes, so we can combine the experimental data and the simulations for modeling time diffusion on free energy landscapes. And then at the end we can say something about the molecular events that are taking place. So here's the synthetic data. We do temperature jump, small angle X-ray scattering in the computer, ignore the timescales. This is just to show that we can generate synthetic data that looks realistic, but we know exactly what's going on because we generate it by simulations. So this is the data that we will interpret, and it corresponds to unfolding of a large complicated protein. So this is again like this is the native state. We pretend that we do a temperature of experiment, so this will be the free energy landscape, and the protein will unfold in a non-equilibrium fashion. So we can run molecular simulations, we can calculate the free energy landscape at the new conditions, like the temperature jump conditions. And now we want to model the process of what is the T-jump SACS experiment actually proving. Right? Again, with the idea is that the SACS data will come with the timescale information and some structural information, and we'll use the simulations to fill in all the structural details. So how do we model time dependency if we only have this kind of free energy landscape? Well, that turns out, unfortunately, to be something that physics has solved for us a long time ago. You can do this in many different ways. We do it by saying, well, we know where we start. Right? We start in the folded state over here. This is the free energy landscape right after our infinitely fast temperature jump. And then we can model this as diffusion. This is something we can do in many different ways. We use sort of a Fokker-Planck kind of modeling, where you start with some Gaussian here, and then you let that Gaussian evolve over the free energy landscape. And so this is what the distribution would look like when you start with a Gaussian here. This would be the folded state. It will decrease in intensity here. It will eventually increase in intensity over here. And you can also see there's some stuff going on down here, corresponding to this stuff over here that's sort of high in energy but might correspond to an intermediate state here. So we can model now the diffusion on this landscape in some arbitrary time scale that we just modeled on this diffusion plot, and then we can restrain that information by plugging in the SACS data. And so this is what it looks like. So this is now the time-dependent SACS data, and then these red lines here are the fits, where we first model this diffusion process. And at each time point, we then use this maximum entropy method to refine the conformational ensembles to fit the SACS data. So we're using what we call a time-dependent prior, which means that we have to have some guess of what the solutions are, and we use this diffusion process on the free energy landscape, and then we use that to interpret the SACS data at the different time points. Okay, so this is sort of what is sort of obvious, right? You start with the folded state here, with the unfolded state here. This Q is a fraction of native contact, and you can see here at some intermediate time point here, you have some folded states, some unfolded states, and some intermediate here. This black line here is this ground truth that we knew was there because we made this with synthetic data. And so you can see that obviously we can fit the starting point and the end point pretty well, but we can actually also fit relatively well with these red distributions here, the fact that there is a transiently formed intermediate. It will be very, very, very hard to see directly in the SACS data. But because they are there in the molecular simulations, even if they're there with the wrong populations, we can see them by analyzing this time-dependent data. And you can again see these kind of things here in the RG plots, although this intermediate is basically invisible in the RG plot, but we can see it in the raw SACS data. So again, I'm jumping through a lot of steps here, and you should ignore most of the colors here. The black line again is this ground truth because we made up the data, and the red point here is what we get out from our method, so we can analyze the data after. We can see a nice drop in the population of the folded state, a nice increase in the population of the unfolded state with essentially a perfect fit. And then we can see this transient population and decrease again of this intermediate state that's really populated at a relatively low level, but at a sufficient level that we can fish it out by using the simulations together with the SACS data. So this is sort of an initial model of this, and we're now, as I said, working on doing the same thing with electrodensity maps. And again, if you soak your ligand in, you will see very messy data early on. So we can interpret that with molecular simulations of ligand binding processes that will clearly not be perfect. But again, by merging the crystallography data in this case and the MD data, we can start to see what are the actual processes, not just seeing the starting point and the end point. In the last minute, I will just mention one thing here, and again, thanks to Martin for already highlighting his own work in this area. These kind of processes where what you measure depends on the actual time scales. So this would be things like an NOE, as I said, Fred measurement, all these things that you really cannot calculate from a single snapshot. And the extreme of that, of course, is NMR relaxation data that some of you will be familiar with. It's something that really is not, something that you can read out from a single plot, right? NMR relaxation involves motion, otherwise you don't get relaxation. So there are many solutions to this. One of them is some work that I did during my own PhD many years ago, which is that you cheat you basically take your messy data that includes both structural heterogeneity and motions, and you deconvolute it. And as I said, I'm not a big fan of deconvolution, but this is what people do at the time. And so you deconvolute it into these order parameters that then sort of are sort of a static component, and then you can fit nice ensembles that fit the order parameters. What we are working on now is building on Martin's absurd approach, and since we love the acronym absurd so much, we came up with a method that's called absurder. So it's like absurd, just even more absurd. The idea is that you want to represent your data by ensembles, but instead of having ensembles of structures, there are ensembles of trajectories. And so you run molecular trajectories, you cut out small trajectories, and you calculate properties from these. They don't fit the data because MD is not perfect, and then you do reweighting as I just explained to you, but instead of reweighting structures, you now reweight conformational trajectories, including the time component. And again, this is work by Felix Plummer and Simonis a while ago. So this is sort of the outline of the approach. You run simulations, you do spectral density mapping. We're interested in this case about sidechain relaxation, which is a long, complicated story that I won't talk about. Calculate these as weighted averages where each of these sum runs over now, not over structures, but over trajectories. And now you can calculate relaxation rates, and you can weigh them so that some trajectories get a greater weight, other trajectories get a smaller weight, and so that the final trajectory ensemble that you get out fit the data. And then in contrast to Martin's method, we have this kind of regularization term here, which is one way of avoiding overfitting, and Martin has another very nice approach of doing that. I'm really going to skip the results of this trial. I have time for questions, just to say that we can then really fit relatively complicated NMR relaxation data directly fitting the data, as opposed to trying to put models on top of the data, to try to, for example, deconvolute them into order parameters or these kind of things. So we're really trying to fit something as close as possible to the direct measurements, in this case, the relaxation rates. And so to summarize here, I think we should always remember, right, that anything that is interesting biologically will move. If it doesn't move, it's probably not so interesting. Sometimes we do stuff to things to avoid them move, which is smart if you want to look at them with certain techniques. But then we have to remember, right, that whatever they're doing in biology, they will be moving. And sometimes we can ignore these movements and we can sort of imagine it afterwards. But if the measurement that you are doing is on something that is mobile, it is really important to put it, right? And we heard a little bit about this before. Right? There are, of course, some approaches of taking EM data and using it as a single molecule technique, but I think we're still very far away from being able to do that routinely, so we're still in the world of classification and averaging. There is, in addition to this kind of mess, there is the conformational and compositional heterogeneity, where what you're measuring on has mixtures, and you can sometimes be lucky and deconvolute, sometimes you can not, and in that case you can take this kind of forward modeling approach. And then just a couple of snapshots of what we are working on, trying to put time back into these ensembles, so that we say, well, it's nice to have ensembles, but it's even nicer if we can say something about the dynamics and the motions within this system. So these two examples. I think I mentioned most of the people along the way, so I'll just stop here, and thank you for your attention, and be happy to take questions.